Hi friends, welcome to Classes by Lab Doctor. In today's class, let us discuss about the types of lipoprotein. In the last class, we have discussed the structure of a lipoprotein. So just to brush up, this is a structure of a lipoprotein. It has an outer core and an inner core. So the outer core is made up of this phospholipids and the unesterified cholesterol. So these phospholipids have hydrophobic as well as hydrophilic end. So because of this phospholipid, this entire structure is able to mix in the blood. Otherwise, the inner core having lipids, that is your cholesterol and triglyceride, they won't be able to mix with the blood. So to carry this is a van which carries this, and it is forming a coat or a membrane here, and in this pocket it is moving in the blood. So there are apolipoproteins also which provide structural integrity and also help in the docking of this lipoproteins to where it has to reach. And these are the lipids here, cholesterol and the triglyceride. So now, how do we classify these lipoproteins? So lipoproteins we can classify on various bases. One is on the based on their density. Second one is the electrophoretic mobility, and third one is the type of apoprotein which is present in that lipoprotein. Now, based on the density. Now, what is density? You can see this one. Suppose if this is an iron nail, if I think, then you can see the particles, they are so closely packed to each other. There is no space between them. So this is high density material. Whereas here the particles are loosely packed together. There is more space in between them. So this is low density. So based on the density, how tightly packed it is, based on the mass, we classify them as a very low density, we classify them as intermediate density, low density and high density lipoproteins. This is based on the density. So this is the, you can see the chylomicron, the density of this is less than 0.95. Whereas you can see HDL, it is having the density of 1.063. So this is highly dense. Whereas this is very less dense. There is enough and more space here. Whereas this is closely packed. So this is high density. Then you can see LDL, low density, intermediate density, very low density, and this is the chylomicron. So based on the density, and this is the proportion of cholesterol here, triglyceride here, phospholipid here, and apolipoprotein. So you can see that in the chylomicron so much triglyceride is there, and if you see here in HDL, half of it is the protein and the rest are your cholesterol, triglyceride and phospholipid. This is what I was telling you, that it is the classification based on the density, the good and the bad cholesterol. So let us study about this high density lipoprotein. So here what you will see that this high density lipoprotein has around 30 to 35% of proteins in it, and this is a good cholesterol. What we say, HDL is a good cholesterol. Why is it good cholesterol? Because it will collect fat molecules like phospholipid, cholesterol, triglyceride in the cells of the body, and this will take it to the liver to get it broken. So that is why it is called as good cholesterol, because higher concentration of this HDL will reduce the risk of having atherosclerosis. So we want the HDL, the good cholesterol, to be high in the blood. So this is the structure of HDL. Here you can see the lipoproteins here. This has more of proteins. Half of it almost is protein here. And in the cardiovascular system, it plays an important role. It protects the endothelium. It has antioxidative activities, antithrombotic activity, vasodilatory activity. That is why it is very important for the heart health. So this is HDL. You will see that it is very small. The diameter is only 5 to 15 nanometer, and it is having around 33 to 35% protein. And the next is your LDL cholesterol. So this is the low density lipoprotein. So this LDL, this again in healthy individual, it is composed approximately about 25% protein and about 21% phospholipid and around 4% of triacylglycerol. It is made up more of this cholesterol here. So the cholesterol particle is more. This LDL is important for the transport of this fat molecules such as your lipids like phospholipid, cholesterol and TG. It is sometimes we call this as a bad cholesterol because if you have an increased LDL cholesterol, that it suggests that you might be at an increased risk of developing atherosclerosis or cardiac disease. And there are two subtypes of LDL, that is large LDL type and small dense LDL. So this is small dense LDL and this is large LDL. So the functions of LDL are that I told you this is the main carrier of cholesterol. So it transports cholesterol from the liver to the peripheral tissue. When the liver, you know that it is a major organ which controls the cholesterol synthesis. So whatever cholesterol is there, when it has to be transported to the various peripheral tissues, it is carried with this kind of a lipoprotein, that is LDL lipoprotein. And LDL are an important source of cholesterol to the extrahepatic tissue. Other than your liver, if you want cholesterol anywhere, then it will be carried by this van, that is the LDL van. And I've told you that this is a major transporter of LDL. It is a major transporter of cholesterol, and the liver plays a very important role in controlling this LDL cholesterol because most of these LDL receptors are present in the liver. So this liver synthesizes cholesterol. It is a factory. It removes cholesterol from the lipoprotein remnants. It breaks down the cholesterol from this LDL, and it is the only organ that can excrete cholesterol through the bile. So liver is an important organ which is handling this kind of a lipoprotein. So here you will see that the LDL cholesterol. So this is the LDL cholesterol. Here you can see this part. The cholesterol is very high, has a very small amount of free cholesterol. Cholesterol esters are very high, and a small amount of triglyceride, and these are the protein component. Next is your intermediate density lipoprotein. So in between, intermediate is in between the VLDL and LDL. This is an intermediate product, and this one is not usually present in the blood when we are fasting. So this are formed during the metabolism of VLDL, and they play an role in the transport of cholesterol. With some intermediate density lipoproteins, they are taken up by liver, or they can be converted into this kind of an intermediate density lipoprotein, or it can be converted into a low density lipoprotein. So this you can see here, this is chylomicron VLDL, this is the chylomicron remnant, and this is an intermediate product between VLDL and LDL. And when I see this is ideal, you will see that this part, this is a cholesterol ester, and here you can see that this is a free cholesterol. This is also increased here, and here the triglyceride part is increasing here. So this is the ideal, and this is the small protein part. Protein is highest in HDL here, and the least here you can see there is no protein at all. So VLDL is the main transporter of this triglyceride, and whatever liver synthesizes this triglyceride, this is carried with this van VLDL to the adipose tissue for storage. So VLDL is very high in triglyceride. That is why the increased amount of VLDL carries a increased risk of atherosclerosis. VLDL, it has a diameter also about 30 to 80 nanometer. It is having around 50% triglyceride. So 50% of triglyceride is carried by this van VLDL. The rest are proteins and cholesterols and phospholipid, but 50% of TG is carried with the help of this VLDL. And then comes the chylomicrons. So chylomicrons are the one which are responsible for transportation of triglyceride in the gastrointestinal tract to the other parts of the body, such as liver, skeletal tissue and adipose tissue, etc. So when you have to transport this TG, this chylomicrons and VLDL, these are the two main vans which are available. And this chylomicrons, what they do is they primarily transport the dietary TGs and cholesterol from the intestine into the bloodstream. Whenever we eat, and from the food, whatever triglycerides or whatever cholesterol are broken down, they will be transported from the intestine into the bloodstream and then to the various tissue, like it has to be carried to the adipose tissue, muscle tissue, liver, etc. That is done by these chylomicrons. They also act in the hydrolysis of TG. So as chylomicrons circulate, the lipoprotein lipase, this enzyme, what it will do, it will break down the TG, it will release the fatty acid and also the glycerol. So this glycerol can be used as energy and the fatty acid will be stored in the body. So once the significant portion of triglyceride is removed, the chylomicrons become the chylomicron remnants, which are taken up by the livers, remnants. So chylomicrons, they are very large, and they have around 80% of triglyceride. So you can understand how much triglyceride is here and how dangerous it can be, and this is one of the major vehicles along with VLDL to carry the TG, and it has very less amount of proteins. So that is about the classification of lipoproteins based on the density, high, intermediate, low density lipoproteins. Now you can also classify the lipoprotein based on the electrophoretic mobility. That means how do they move when we place it or when we do an electrophoresis. So those lipoproteins with minimum protein content will move faster, and those lipoprotein with higher protein content will move slower. So the movement of lipoprotein is based on the nature of the apoprotein which is associated with the lipoprotein. So there are alpha lipoprotein which are having the highest protein content. HDL are the alpha lipoproteins. VLDL are the pre-beta lipoproteins, and LDL are the beta lipoproteins, and IDL are the broad beta lipoproteins. So the lipoprotein with minimal protein content, that is a chylomicron, this is the one which is going to move very fast, and the HDL, which is having the highest amount of protein, this is going to move very slow. So this is the alpha lipoprotein, that is the HDL, and this is the pre-beta lipoprotein, VLDL, and the beta lipoprotein, this is the LDL. So depending upon the protein content, the movement will vary, and according to this also we can classify them as alpha lipoprotein, pre-beta lipoprotein, beta lipoprotein, and chylomicrons. Other classification wherein we classify the lipoprotein based on the apoprotein content. So what happens is, this apolipoprotein is a very important part of this lipoprotein. This is known as the brain of this lipoprotein system, because they regulate the remodeling, the uptake, the docking, the lipoprotein complex formation, ligand formation, etc. So with the help of specific enzymes or proteins that complex binds to on the cell membrane, the lipoproteins are directed to the metabolic site. So this direction is given by this apolipoprotein. So these apolipoprotein, we broadly classify them into A, B, C, and E. HDL is the one which consists mainly of apolipoprotein A, while LDL and VLDL, they consist mostly of the apolipoprotein B. And the chylomicrons, which have the truncated form of apolipoprotein B, that is ApoB48, and apolipoprotein C occurs in HDL, VLDL, chylomicrons, and apolipoprotein E occurs in all the classes of lipoprotein. So we will study this in detail again, but this is just to give you a brief idea as to how we classify the lipoprotein. We can even classify them according to the apolipoprotein present. So here you can see a chart where you can see the different types of apolipoprotein present, and we can classify these lipoproteins according to the apolipoprotein present also. These were the various classification of lipoprotein based on their density, based on their electrophoretic movement, and based on the apolipoprotein present. So in the later classes, we will dissect each of these components, and we will try to learn in detail about each of the lipoproteins. Thank you so much for watching the video. Please like, share and subscribe the channel. Thank you.